name is Jason, and I'm currently an assistant professor of biostatistics and bioinformatics at Duke University School of Medicine. Um, so my lab is developing methods and computational statistical methods for analyzing genomic data, particularly from single cell spatial transcriptomics and biomedical imaging. And right now, I co-lead the data analysis call with Dr. Cleben Chen of the Duke Tissue Mapping Center for SenNet. So we actually when we started to work on the SenNet project, so one of the major goal is to identify the senescent cells in the human tissue. So Duke is focused on lung, normal human lung and colon. So we did a survey of methods that can identify senescent cells. So there are both methods that are general purpose, so just methods that can identify any kind of specific cell type, and methods that have been specifically designed for identifying senescent cells. So we did a small like benchmarking study by ourselves using data collected online, so published data from other labs. I found that some of the methods, although it worked well in certain cell types and tissues, it did not work well in others. So that means the method is not generally applicable. And we tried to reason why that's the case. So take one method. There's a method called SenSet as an example. So that method is actually a so-called supervised method that's trained on a training set and then tries to apply this trained model on a testing set. But we noticed that it was trained on cells that are collected in vitro, that cells are collected outside of the basically grown in the labs. But when we apply it to the actual cells in the human tissue, we noticed that there could be a huge difference between cells grown in lab and cells in human tissue. So that means such kind of method may not be applicable in our setting. So we thought that probably it might be useful for us to develop our own method that can be generalized, generally applicable, and can be applied to human tissues. So that's why we try to think of the idea of developing a more general method. And because of the lack of the training data, we sort of why don't we take a different approach? Instead of using supervised training method, we use the unsupervised method. So this kind of thinking has been around for some time in the single cell studies. Other studies have also used this, but instead of just applying to the whole gene expression information, we only applied this kind of methods, subset of genes that we know to be associated with senescence. So we collected such kind of a gene set, like 30 or around 40 genes that are associated with senescence by surveying a set of publicly available gene sets. And we developed this method based on this gene expression only for these around 40 genes. And it turns out that the results is pretty good and it can be applicable to many different cell types and tissues. So that's why we saw that this kind of method, we call this is based on unsupervised training, that we don't specifically have a training set, and this basically is tries to aggregate the gene expression of genes that are, so if you have 40 genes that are associated with senescence, basically the method tries to aggregate the information and gives you one score that reflects the senescent state of the cell. Although it's not a simple averaging, it's kind of based on some deep learning method, but the idea is that we aggregate the information of senescence-related genes. And that score can be used to, for example, tell which cell is senescent cell, which cell is not, and later can be used to compare cells, senescent and non-senescent cells. So that's a basic idea and motivation behind this method. In terms of how the DeepScence changes what we are doing within SenNet in general field of cellular senescence, I think the so if we want to study the senescent cells, we first need to identify them. We first need to find what cells are senescent cells, what cells are not, before we can compare them. So identifying senescent cells is crucial, and being accurately to do so also reflects the reliability of the conclusions we can have. So, although the previous methods can also identify senescent cells, as I said, in our benchmark, we found that they may not be applicable to many different tissues. So, one advantage of DeepScence is its generalizability. So, it can be applied to different kinds of tissues, cell types. We all have benchmarks for that in our paper. It can also be applied to different kinds of um, sequencing or profiling platforms, including single-cell seq, different kinds of spatial transcriptomics, including Visium, Xenium, and other kinds of similar platforms. One difference between these spatial and single-cell profiling technology is that the spatial data usually only contains, in some cases, only contains hundreds of cells, hundreds of genes, sorry. And a single-cell seq gives you basically all the information of all the genes of the cell. So our method is flexible in terms of the number of input genes, number of the genes you profile for a cell. We have also tested the previous method, and some methods just does not work if you only have hundreds of genes in your data set. So basically, DeepScense gives us a chance to study senescent cells in many more tissue cell types and for data coming from different platforms, while previous methods probably is more confined to specific cell types, tissues, and sequencing platforms. So that allows us to gain broader knowledge of the senescent cells in different scenarios. As I said, the DeepScence method is quite general. So that means it can be applied to both previously published methods and data set studies and data sets, and also other data sets are being generated. So as long as it's a single cell seq or spatial data set that contains some of these senescence gene markers, um, although maybe the previous, like the original goal of this study data set is not to study senescence, it's for other purpose. But as long as the data set is there, then people can use this DeepSense and the related downstream analysis to study, to kind of repurpose this data set for senescence study. So that gives us, it's a potentially a huge resource for us to study the cellular senescence in different kinds of tissue, cell types, disease status. So again, I think current knowledge of cellular senescence is in general limited. So there are studies that are particularly focused on cellular senescence, but those are like based on some particular cell types, tissue types. So if there's a method that can generally applicable to all kinds of data sets, then it's possible that people can gain a lot of knowledge from these previously published data sets that are coming from other tissue and cell types diseases. Right now, I think DeepSense is a method that's only based on gene expression. And this senescence associated genes we collected are from previous database. And these databases are more or less focused on specific cell type. I think it's mostly epithelial and some of them are fibroblast. So we can call this a phenotype. It's type of cellular senescence, but there are definitely other types of cellular senescence that is not that widely studied in the literature. So in the future, I think there's several directions to pursue. First is that one can rely on other type of data modalities such as images, cellular morphology information to tell the cellular senescence. In addition, or instead of gene expression, depending on the availability of the data. And second is that we probably need to collect information about other phenotypes, not only like epithelial and fibroblast centered phenotypes, but other kind of phenotypes to to be able to study cellular senescence more accurately in other tissues. So I think there are still a lot of work we can do to push this field forward. Yes. So some of my experience in thinking about SenNet, I think SenNet is a major like a resource and it's a maybe it's a first consortium to systematically study cellular senescence in normal human and probably diseased human tissues. I think this SenNet is very essential for pushing this forward. Previously, I think the effort in cellular senescence is more like carried forward by individual labs, but there's no like a systematic effort and organization of pushing this whole field forward. And also, take myself for example, I did not have any experience in cellular senescence before I joined SenNet. So I really appreciate SenNet for providing such an opportunity for like early career scientists and other scientists who do not have experience in cellular senescence to introduce them into this field so that they can also contribute to this very important field. I really think cellular senescence is very important, but I didn't I didn't have any knowledge in that before I joined SenNet. So it's also very educational for me and introduce a very new and exciting and important research direction, um, which is I think it's very important because it's linked to human health, human aging, and many different kind of age-related diseases. And right now, I think our knowledge is still limited before we can really find for example, a treatment to slow down the aging or even reverse the aging process or treat some of the age-related diseases. But I think SenNet is crucial in collecting the knowledge and developing the essential technology to gain this to make the step forward so that we can really um, move closer to the clinical and more like treatment diagnostic type of um, um, treatment of these age-associated diseases. So yeah, I will briefly introduce the first author of this paper, Anthony Chu, who was a master student and research staff in my lab, and now currently a PhD student at University of Pennsylvania. So he actually came up with this idea of using this kind of unsupervised learning to identify senescent cells in a broader context. And he actually developed the whole software and performed this data analysis, um, and partially contributed to the writing of the manuscript. So he is really the driving force of the whole manuscript. Of course, we also have received a huge contributions from within the Duke SenNet consortium, from the UW Technology Development Center, and also broadly from the SenNet consortium and people outside SenNet. So the success of this project is really I really want to thank my student and other people for making this possible.
Hello, SenNet team. My name is Anthony Q. I am a graduated master's student from the Duke University, where I worked in Dr. Jason G's lab on senescence and SenNet related research. Currently, I am a first-year PhD student at University of Pennsylvania. This video gives a quick tutorial about the package DeepScence that I developed, which was my master's thesis project. Here I'm showing in the screen is the GitHub page of the DeepScence package. This package takes as input a and data object, which is either single cell dataset or spatial transcriptomics dataset that you have. And what it does is append two columns in the metadata data frame of the dataset, which is a continuous senescence score, where higher scores means more senescence, and a binary classification of each cell. And then which is column binary, it takes on values senescent cells and normal. The first step is I'm going to show the installation of the package on my local computer right now. So the installation has two options, and because the installation pipeline will be different depending on the operating system that you're using. For example, if you are using Linux machine, for example, on computer clusters in your institution, you may go ahead and look at this section right here and follow each step to install DeepScence. On my computer right now, I'm using Mac OS. So for Apple users, you will go ahead and look at the second section of the installation tutorial and follow these steps. So I'm just going to follow these steps right now on my terminal. And so first of all, we create a kind of environment with Python version 3.8, and we'll go ahead and activate this environment because I renamed it to test. So I'm just gonna activate this environment, and the third line is a checking, where the expected output is going to be ARM64. This is just a configuration that that makes sure the later installation will go smoothly. So the next step would be just copy and paste these three steps, which installs DeepScence, TensorFlow, as well as the package DCA, which is required for later steps. Okay, so that is the installation. You can see that it installs just fine. And within this Python, within this kind of environment, you can go ahead and perform all the senescence analysis with DeepScence. So the second part of the tutorial, I will go ahead and show you how to use DeepScence in a real data set. So this tutorial Jupyter Notebook is available on the GitHub page as well. But right now, I'll just run it on my local machine to show the expected output and the parameters to use. So as an introduction, there are two types of dataset that we used in the paper as a benchmark. The first type of data is in vitro single-cell RNA sequencing datasets. And the reason we use this is because the in vitro dataset that we collected actually had a golden standard of senescent versus non-senescent labels for each cell. And also in vitro cell line dataset, it would only normally have only one cell type. And this is a good validation of DeepScence and its accuracy and performance. So the first step would be import necessary packages. For a standard DeepScence analysis, you would use ScanPy, NumPy, and just plotting a bunch of plotting packages that you use for checking the results. So after importing the packages, this section gives an overall introduction of DeepScence package and what it does. We can jump right into the example dataset, which is again the in vitro IMR 90 cell line single cell RNA sequencing dataset. This dataset, a little bit introduction, is that part of the cells come from a sample which was induced senescence by OSKM factors. And this information was recorded for each cell so that we know the ground truth information for each cell, whether it's a senescent cell or not. And the first step would be constructing the ScanPy and data object for this single cell RNA sequencing experiment. We read the count gene expression count matrix from the GitHub tutorial subfolder. This is also available on GitHub as well. And we construct the AND data object. And the senescence information in this dataset was recorded in the cell names. And we'll go ahead and set senescent cells as those cells that have been treated with OSKM factors. And after generating this entire dataset, this single cell experiment dataset, we will go ahead and do a pre-processing step, which is the first step is removing all the genes that have zero expression across all the cells. This step is required for the next step, which is DCA. This is a denoising step that will impute the gene expression count matrix. So we go ahead and run this. The expected output would be DCA's output. It says it's processing a certain number of genes across a certain number of cells, and this is rather small data set and it doesn't take long. And after you have a denoised and data object, the next step will be run DeepScence. And DeepScence is a wrapper function that contains all the pre-processing, model training, and output generating steps. So all that you have to do is to run this line of code where it says DeepScence takes in as input the AND data object, and this binarize equals true parameter basically says that you want to binarize the cells into senescent cells versus normal cells. And we'll go ahead and run that. DeepScence first of all, the first step is that DeepScence detect whether GPU is available on your machine. If not, go ahead and use CPU. If it does, it can accelerate the computation by using GPU. And this are just some recording diagnostics. And what it does, when you set binarization equals true, you will have two diagnostic plots. The first plot is the distribution of DeepScence score, continuous score across all the cells. And the two density curve is actually drawn based on the known senescence labels. And this plot can only, this density curve will only appear when you actually have the senescence information in the dataset. But most of the time we actually don't. But as a validation purpose, you can see that senescent cells or the cells treated with OSKM factors received a higher DeepScence score, and normal cells had lower DeepScence scores. And the second diagnostic plot shows the binarization procedure of DeepScence. What it does is that given the same distribution of DeepScence scores, it fits a mixture of two Gaussian models and identify the cutoff being a certain certain score in the continuous score, and whatever cells that received a higher score than that will be identified as senescent cells. And just to check the accuracy of the above results, because we already know the ground truth, we go ahead and plot for two things, which is the AUC curve and the confusion matrix for classification. So after plotting, you can see that normal cells, senescent cells, received a significantly higher DeepScence score comparing to normal cells, and the classification was pretty accurate in this dataset, reaching an accuracy of 94.5%. So this is a validation with in vitro dataset, but then most of the cases when people study senescence, you would actually look into in vivo dataset instead of in vitro, because in vivo gives you just so much information. And then, but one main difference or main distinction between in vivo and in vitro dataset is that it's much less homogeneous, where in vivo dataset you would have multiple samples and multiple cell types. When we run DeepScense, we want to control for these factors so that we avoid the DeepScense model to be confounded by variations that are not based on senescence. So here we introduce a second example dataset, which is a mouse muscle dataset. This is a multi-sample as well as multi-cell type dataset where a subset of samples were treated by cardiotoxin, and senescence in the muscle cells were induced by this cardiotoxin. The paper was able to show that this treatment results in higher beta-gal staining as a orthogonal validation of the senescence induction. So what we do with this data set is, first of all, we read the data set into scanpad data object, and still we filter the genes that are expressed in 0 cells, and then we perform the denoising step with DCA as before. This is a rather large data set, so the DCA will take some time, maybe a couple minutes. Okay, after having the data denoised with DCA, we will, in this case, we will have one additional step for all in vivo dataset when you're using DeepScence, is that we want to control the variation that arises from cell type difference, because we want to focus on the variation from senescence or normal instead of the cell type. So what we do is we will go ahead and set this column of a data observation to this column B stands for batch, as whatever column in the observation that you want to control for. For example, here you want to control for the variation arise from cell type. You go ahead and set a data.observation column B equals the cell type. Sometimes people also want to control for biological replicates, samples, or samples and cell type at the same time. So what you can do is you can just create pairs and assign into batch column. For example, if you want to control for cell type and sample, you would do a data.obs.b equals cell type plus sample. And then after having this this B column in the observation data frame, you can go ahead and run DeepScence. The package will automatically detect that you're trying to control for cell type, and it will run a version that actually controls the variation of cell type, so that we can focus only on the information from senescence. And this data set, remember this data set is mouse muscle data, and mouse genes and human genes have different nomenclatures, different gene names. So when you use DeepScence on mouse data set, you might need to set species equal to mouse so that it can actually reason mouse genes. The default value for species human, if you don't set anything, that means the model assumes that you're dealing with human genes. Right now, the model only supports mouse and human genes. So we go ahead and run DeepScence on this data set. Again, we have the similar set of outputs. And for this run, I set binarize equals to false, because in this dataset, in this in vivo dataset, we actually do not have ground truth information of each at cellular level. Basically, we don't know each individual cell whether they're senescent or not. But we do know what we do know though is we know the condition, um, the samples where come the cells where the cells that come from the sample induced by cardiotoxin, these cells should have a higher enrichment of senescent cells, thus a higher overall distribution of senescent scores. So we go ahead and plot to check if this is true. We plot a box plot where the x-axis are cell types. We can see this is a multi-cell type dataset, and the y-axis is the DeepScence score. And each bar is colored by the biological condition of the cell, whether it's coming from control or the samples treated with cardiotoxin. And we can see that in every cell type, the cells that were treated with cardiotoxin received a higher DeepScence score, meaning that they have a higher enrichment in senescent cells in compared to the control samples. So that is basically the general use case of DeepScence package. There are a few additional parameters that you can set when you work on your own data set. For example, when you work on mouse data set, like the one above, you go ahead and set species equal to mouse so that it can recognize mouse genes in the DeepScence model. And also in some cases, so the DeepScence model uses a gene set that we call coresense to identify senescent cells, to score and identify senescent cells. The coresense is a set of genes that we believe is consistently associated with cell senescence across many tissues. And sometimes when you are working on your own data set, you might want to test another list of genes. For example, the list of genes that you think is associated with the senescence, particularly in your own tissue or organ or cell type, because senescence is so heterogeneous. So if that's the case, you can go ahead and set gene list equals to a list of genes that you think is important in your data set. And then when you run DeepScence, you will go ahead and set custom GS, which is custom gene set, equals to gene set, which is a list of genes that is pre-specified. In this case, DeepScence will go ahead and score and identify senescent cells based on the expression of the genes that you provided. So that's pretty much it. Thank you for listening.